Hello everyone. It gives me great pleasure to present to you the educational video presentations on cancer, particularly with regard to the basic biology of cancer. And over a period of time, we will have more titles covering different aspects of cancer, moving on to more advanced topics on cancer as well. I hope this would help the medical fraternity, medical students in particular, postgraduates, biotechnology students, and maybe a few faculty members as well. I'd be happy to receive your comments and thank you. And I hope that this is really useful to you. Thank you. So, we all started life as a single cell zygote following the fusion of the sperm with the ovum. This then gives rise to the early divided cells, which actually can give rise to a full individual, each one of them, which is called as totipotent. Subsequently, there is more division and some more differentiation, making these cells give rise to specific elements of tissues. So these could then be giving rise to the blood cells, to the skin cells, to the muscle, bone, cartilage, connective tissue, etc., and also to the nervous system. Now, all this is going to need both proliferation and differentiation. And at the end of the day, this will then result in a full grown fetus will be delivered by the mother and develop into an adult. Now, what happens if the zygote does not divide? If there is no proliferation, it stays put as a single cell and would die after some time. If, however, there is only proliferation but no differentiation, we would just be a bag of cells. So, how do we define cancer? So, cancer can be defined as an uncontrolled proliferation of cells which are no longer under the normal regulatory control and also acquire the capacity to invade into neighboring tissues and spread to distant organs. So, I've given a small cartoon here which shows you the differences in the way the cells in the normal compartment behaves and how the cancer cells behave. Let's presume that in the normal, this is possibly a skin. So at the basal level of the epidermis, we have stem cells. These will undergo proliferation, asymmetric kind of division, meaning that the two divided cells will not have the same properties. One of them will continue to be stem cell. It is the other one which will continue to enter into the proliferative compartment, give rise to a large number of cells. Now, once they have divided to an extent, then differentiation comes in, which allows these cells to be fully differentiated to carry out specific functions. And in the case of the skin, they protect, and subsequently, these cells are shed after they are died. And this will also require this process to be continuously repeated so that the skin integrity is maintained. In contrast, if you look at the cancer cells. So these cells proliferate, have stem cells, cancer stem cells also, and these for the predominant kind of proliferation, as you see here, you find the differentiation becomes much less, and the few cells which are differentiated may be there. There are also cells which are going to be dying. They die possibly because of too large mutations which the cell is unable to bear and therefore dies, or it may be that the death is occurring because there are short of nutrients. Either way, you find that there is a complete disorganization of the tumor, the way the cells are organized. They are not in an orderly fashion as well. Coming to the controlled proliferation examples, I would give wound healing as an example here. You need to have replenishment of the normal tissues after an injury so that that area is the cover is retained. like in the case of a cut on the skin. We also have continuous replacement of cells needed in the GI tract, in the skin, in the bone marrow, the uterus lining, for example, during menstruation, etc., which is necessary to maintain the integrity of all these tissues. However, uncontrolled proliferation can occur both in malignant tumor and benign tumor. So, how does a normal cell become malignant? A normal cell is being always bombarded by a wide range of carcinogens, every one of us. So, these carcinogens or cancer causing agents can be of chemical nature, physical nature, or biological agents, which we'll see a little later. The target for these essentially is the DNA damage. DNA, as you all know, carries the genetic code for the individual. Now, the DNA damaged cell is given an opportunity to repair the DNA damage. There are mechanisms which will be triggered, which will allow the DNA damage to be repaired and the cell can then survive. If, however, the cell fails to repair the DNA damage, and then another set of protective mechanism called as induction of programmed cell death or apoptosis comes in. This will lead to cell death. The cell which had the DNA damage will die, thereby there is no further risk of cancer. In all of us, we have millions of cell division happening at any point of time. And there is always a possibility that something can go wrong in some of these divisions. And it is these protective mechanism which prevents us all from developing cancer. In the event that the cell does not repair the DNA damage and these protective mechanisms fail, then it starts accumulating more and more DNA damage, leading to what we call as activation of oncogenes and inactivation of tumor suppressor genes. And this can then lead to cancer. And following acquisition of more number of genetic changes, which can lead to spreading to other distant sites, it will lead to the death of the individual. Among the carcinogens, the tobacco-related carcinogens are very important. And tobacco smoke, for example, contains more than 7,000 chemicals, of which nearly 70 of them can cause cancer. And these are a wide spectrum here, including benzene, benzopyrene, etc. And then we also have heavy metal like cadmium, chromium, and then we also have polonium 210, which is a radioactive compound. So all of these are likely to cause cancer. Tobacco-related cancers include lung cancer, head and neck cancers, particularly oral cancer, pharyngeal cancer, laryngeal cancer. And then we also have tracheal and bronchial cancers, esophageal cancers, pancreatic cancers, kidney cancers, and bladder cancers. And that's the urinary bladder cancer. Several occupations are also having certain risk due to the chemicals or the way the occupational industry works, which can predispose to cancers. So mining, for example, is associated with a wide range of cancers, including particularly lung cancer, like mining for arsenic or asbestos or beryllium, for example, here. Chemicals like aniline, which were used in dye industry, can cause urinary bladder cancer. And then we also have benzene used in rubber and cement workers and dye users, which can cause blood cancer, the acute myeloid leukemia. We also have beta naphthylamine, which can cause urinary bladder cancer. As I mentioned a little while earlier, the miners, like for hematite mining, nickel mining, uranium mining, are all predisposed to cancers like lung. Cadmium, again, is another risk for lung cancer, prostate, and kidney as well. Wood dust can predispose to nasal cancers. Pesticides used in farming can predispose to lymphomas and lung cancer as well. Having said this, several regulations have been brought in to minimize the risk to workers in the different fields. And therefore, Currently, a large number of these regulations have helped save the lives of several people. Drugs which are used to treat diseases can also cause cancers. Among these are the anti-cancer drugs, the alkylating agents which can cause acute myeloid leukemia, a form of aggressive blood cancer, anabolic steroids used by some of the sports or people can actually cause liver cancer. Immunosuppressive drugs like cyclosporin and azathioprine can cause lymphomas, skin cancers. Diethyl stilbosterol, which is used in treating threatened abortion, can unfortunately cause vaginal or cervical adenocarcinoma in the daughter. Tamoxifen is a drug which is very widely used in the treatment of breast cancer, and unfortunately, it also runs a risk of causing uterine cancers. Among the physical agents, we do have ionizing radiations, UV radiation, and mineral fibers, all of which we'll see shortly. Ionizing radiations will include electromagnetic, which are X-rays and gamma rays, and then we also have particulate ones like electrons, protons, alpha particle, uncharged ones being neutrons. The X-rays and gamma rays, interestingly, are also used in therapy or for diagnosis, and unnecessary use of these X-rays, especially for diagnostic purposes, runs the risk of developing cancer. More often than not, radiation is quite effective in producing DNA breaks, deletions, and chromosomal translocation, and thereby leading to possible cancers. UV light, especially from the sun, has a wide range, of which the ultraviolet beam, particularly between 280 to 320 nanometers, is most effective in inducing DNA damage. The form of DNA damage is quite unique in that it can cause what is called as pyrimidine dimers. So, what are pyrimidines? In the DNA, we have four alphabets: adenine, guanine, cytosine, thymine. Now, adenine and guanine are purines, cytosine and thymine are pyrimidines. When in the sequence we have two consecutive cytosines or two consecutive thymines, these regions are particularly prone for UV induced damage. The UV, as you see here in this cartoon, causes covalent linkage between the two adjacent thymine, which actually, as you see, it distorts the DNA structure, making it very difficult for DNA replication and transcription. So both the cell the division or the DNA proliferation as well as also mRNA transcription gets affected, and this therefore can result in skin cancers. Another physical agent is asbestos, and the important ones are the crocidolite and amosite, which can cause several deletions. And this is particularly important for a cancer called as mesothelioma, which is the cancer in the lining or the outer covering of the lung, as well as also within the abdominal cavity, the peritoneal cavity. It can also cause lung cancer and possibly kidney, pancreas, colon, and esophageal cancers. Among the infections which can cause cancer, we have Epstein-Barr virus, which can lead to different types of lymphomas as well as nasopharyngeal carcinoma. The hepatitis viruses B and C can cause liver cancer. Human papilloma virus can cause cervical cancer and also anal and skin cancers and oropharyngeal cancers as well. The human T lymphocytic virus can cause leukemias and lymphomas. The HIV-1 human immunodeficiency virus can cause lymphomas and Kaposi sarcoma. This is in addition to causing AIDS. Parasites such as Clonorchis sinensis can cause bile duct cancers, cholangiocarcinoma, and schistosoma hematobia can cause urinary bladder cancer. So, what are the mechanisms by which cancer arises? So, the tumorigenesis pathways. So these can be classified as activation of proto-oncogenes, inactivation of tumor suppressor genes, and alteration in the immune response. I'm not going to deal with the alteration in the immune response in this video. We'll try to do this in a future one. I prefer to give an analogy of comparing the proto-oncogenes and tumor suppressor genes to the way one needs to drive a car. If you want the car to move forward, you need to take your foot off the brake pedal and step on the accelerator. Similarly, if you want to cause a normal cell to become a cancerous one, you need to activate the proto-oncogene into oncogenes, and the tumor suppressor genes needs to be inactivated. That is, the brake within the normal cell needs to be inactivated. I've already shown this. I'm just trying to reiterate that there is proliferation and differentiation, both are important. And if you do not have one or the other, it can lead to problems. The proto-oncogenes are not within our body to cause cancer. Proto-oncogenes, as already I pointed out, they have very major functions, allowing a single cell zygote to actually develop into an adult human. You need so many billions of cells to actually give rise to an individual. And that is something which the oncogenes do. However, as far as cancer goes, the normal regulatory pathways for these proto-oncogenes is lost. And as a result, the oncogenes function inappropriately or excessively. How does this activation happen? So this can happen through mutation, gene amplification, increased transcription, deletion of a regulatory element, chromosomal translocation resulting in a novel oncogenic proteins, and insertional activation. We'll see examples of each one of these. So we have the process by which the mRNA is translated into proteins is by reading the genetic code as triplets. So this is why it's called the triplet codon. And I've just given an example here. So TGG, the three bases, when they occur as a triplet, this will code for tryptophan. TCG, when you change the G to C, the central base, it becomes serine, a very different amino acid. TTG, again change from G to T, gives rise to leucine. And if I change the G to A, the TAG is a stop codon. A stop codon means that there is going to be prematurely the translational machinery comes to a stop. As a result, the mRNA is not fully translated and the protein is a shortened version, and most of these do not have proper function and usually they are destroyed. We all receive two copies for each gene. One of them comes from our mother, one comes from our father. Now, in some situations and in some cancers, one of these copies can actually become amplified. So 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. As a result of this is called a gene amplification. You would end up with larger number of the gene copies, which means there is more material of the gene product, meaning there will be more protein synthesized. One example of this is the HER2 or COB2 protein present in breast cancer, and it's it's one of the cause for an aggressive behavior of these breast cancers as well. The next mechanism is deletion of regulatory elements. So I've taken the example of a TAL gene. Now, within our body, every gene has its promoter. It is binding, the transcription factors can bind to the promoter, get the go signal, and then they move to transcribe the DNA into RNA. So you get the mRNA synthesized. Now, again, not all genes are functional in a given cell at all the times. Now, the SIL gene, for example, is what we would call as a housekeeping gene, meaning that this gene has a regular function in the normal cell, and its function is usually in the endoplasmic reticulum, wherein it's supposed to monitor the health of the protein folding. Now, the TAL gene, in contrast, is expressed only in a selected cells and at selected time of the life of that cell. An inappropriate expression of the TAL gene can cause problems. Now, there is a peculiar situation wherein due to translocation or losing a bit of the chromosome, the SIL promoter comes in opposition to the TAL gene. Now, as you recollect, the SIL promoter is continuously active, unlike the TAL promoter. This continuously active SIL promoter then drives TAL gene transcription so that you have inappropriate expression of TAL gene product, and this can then cause a T cell leukemia as well. Another form we are going to be mechanism which we are going to be talking about is the chromosomal translocation resulting in novel oncogenic proteins. And the example I've taken here is the Ewing sarcoma, a rare form of bone cancer seen particularly in young children and in adolescents. Now, we have two genes, the EWS gene in chromosome 22 and the FLI1 gene in chromosome 11. Now, the translocation involving 11 and 22 results in the EWS gene breaking off, and actually what happens is this part is called the N-terminal end of the EWS transcript. This has RNA binding protein. In contrast, the FLI1, which is the DNA binding transcription factor. Now, the DNA binding region is this, the C-terminal domain. So, this is where the C-terminal domain is. So, when the translocation happens, we get a chimeric EWS FLI1 gene, which is not present in any normal cell. And here we have the transcriptional activation domain of the EWS fusing with the DNA binding domain of FLI1. Now, the net result is that because this can activate transcription and this goes and binds to any specific or different DNA, the genes, you would then get inappropriate transcription of a variety of gene products, which can then drive to develop Ewing sarcoma. A rare form of activation is insertional activation. So here this is again an example of the c-MYC gene, which is a proto-oncogene. And the avian leukosis virus could actually get itself integrated between the promoter and it drives the c-MYC gene, producing inappropriate expression of c-MYC gene product. So there are a wide range of proto-oncogenes. I'm not going into the details of all this. There are growth factors and their corresponding receptors like EGF, EGFR, COB2, which are all associated at different cancers. BCL2 is an inhibitor of apoptosis of programmed cell death and is associated with leukemias and lymphomas. RAS is a molecular switch, which we'll see in one other video presentation on signal transduction, which allows the signals from outside the cell to pass into the nucleus. And RAS mutations can lead to large intestine and pancreatic cancers. Cyclin D1 promotes entry of cell into cell division, and that is leading to breast cancers and lymphomas. MDM2 is a physiological antagonist to P53, and P53 we all know is a tumor suppressor gene. But again, it's a negative. You need something to keep the P53 also in check. You don't want a situation where the cell cannot proliferate when it is needed. So MDM2 is an antagonist that way. However, in some cancers, there is overexpression of MDM2 or gene amplification, or what we call as there is an enhanced production of the MDM2 gene product, which can lead to sarcomas. Angiogenic molecules promote new blood vessel formation, and these are critical for most of the cancers including sarcomas. Now, a tumor suppressor gene could be inactivated by deletion, mutation, which we have already seen, viral mediated inactivation. The classical example is the human papilloma virus, which produces two proteins, E6 and E7. E6 inactivates P53 and E7 inactivates the retinoblastoma protein. So both of these P53 and RB protein, the tumor suppressor genes. So you have the breaks removed from a normal cell, and a few additional mutations involving the oncogenes can then trigger the normal cervical cell to become a cancerous cervical cell. Functional inactivation by oncogene, which we discussed a little while earlier, where P53 can be inactivated by overexpressed MDM2 and then promoter methylation. Promoter methylation is a very important physiological phenomenon. It plays a very important role since we do not need all the genes in our body to be active at all the times in all the cells. So there are mechanisms by which the genes can be silenced. So here in this cartoon, I've shown a gene and its promoter. The promoter region has sequences of C, G, C, G, C, G. So this is called as the CG or CPG island. So you have runs of CG doublets occurring. Now, the promoter binding transcriptional factors like AP1 or SP1 can bind to the promoter, and then they can trigger transcription so that the mRNA can then be synthesized. Once mRNA is synthesized, it gets into the cytoplasm where the translation happens. However, if you want the gene to be silenced, you can add a methyl group to the cytosine. And this methyl group addition happens at this CPG island only, not when the C is independent or C is followed by a T or A. It has to be the CPG group. Now this methylation will now interfere with the binding of the transcriptional factors, as a result of which transcription is inhibited. So there is no transcription. So there is no gene expression. So physiologically, these are very important mechanisms, but in cancer, this is one of the mechanisms by which the tumor suppressor genes can be inactivated. Again, there are a wide range of tumor suppressor genes. We've already seen P53 and RB gene. P16 and P15 regulate the G1 phase of cell cycle. NM23 is a non-metastatic gene which helps in preventing metastasis. And mismatch repair genes and other DNA repair genes, this is going to be a topic later on that's in a separate video. So they help in maintaining or correcting the DNA damage and helping to repair the DNA. So here in this slide, I'd like to give a little more elaboration about the function of P53, which is otherwise called as the guardian of the genome. Now, as I said earlier, the cell is a target for a wide range of cancer-causing agents, including radiation, viruses, chemicals, UV light, etc. The impact of this is that there is going to be DNA damage. The DNA damage is then sensed, and then this will then induce the synthesis of P53 protein. The P53 protein in turn promotes the transcription of other genes like P21, which can inhibit the cell cycle, GAD and VAX, which can trigger apoptosis, and MDM2, which is a physiological antagonist to P53. So the effect of this is that the P53 is saying to the cells, now stop dividing, repair your DNA, you will then survive. If you fail, if you do not repair the DNA, then you will die, and the death is usually through apoptosis. So once the DNA damaged cell dies, the risk for cancer is eliminated. So here I need to talk about what is called the multi-stage genetic alteration in colon cancer, which was identified by Bert Bergelsen, uh, and here he had actually given the sequence with which a normal colonic epithelium can become malignant. So he showed that the normal epithelium becomes hyperplastic, meaning several layered thick, which is abnormal. The several layer thick epithelium can then become a benign tumor called as early adenoma, which can then progress to become an intermediate adenoma and then into a late adenoma, which is almost a precursor lesion for cancer. Uh, then an invasive carcinoma can result, and with more changes, it can spread to neighboring tissues and to distant organs. So Vogelstein showed that APC gene, which is adenomatosis polyposis coli, which is a mouthful, this gene is mutated. When it is mutated, this is actually a tumor suppressor gene. And this is critical in the stem cell, basal proliferating stem cell of the colonic epithelium. So once you remove this break, the cells continue to divide and result in a hyperplastic epithelium. To move this into an early adenoma or benign tumor, the next set of changes is required, and this he felt was the DNA hypomethylation. A little while ago, we talked about promoter methylation, whereas here we are talking about the opposite of that. So you remember at that time I said not all the genes are required at all the time. So most of the genes are silenced by methylation of the promoters. But in cancer, some of these proto-oncogenes are activated by removing that methylation of the promoter. The promoter methylation is reversed, and that is called as hypomethylation. So this is global, in the sense it is not just restricted to one part of the DNA, but many genes are affected. And this can therefore lead to early adenoma. The next critical element is RAS mutation, which is downstream always of this APC gene in colon cancer. However, in pancreatic cancer, it occurs much earlier. As we already saw, RAS is a molecular switch within the cell which can transduce the cell surface signals to the nucleus. And then the conversion of intermediate adenoma to late adenoma is done through the loss of deleted in colon cancer gene, another tumor suppressor. And then the loss of P53 completes the development into a carcinoma. Now, some of these changes can be occurring at different times, a little earlier as well. Finally, with more additional mutations, you would end up with the capacity of these cancer cells to invade and disseminate to distant organs, causing metastasis. So I would like to thank you for your attention on this presentation, and I hope this was informative, and I hope to see you in our next presentation video, which should happen shortly. Thank you again.